take it away. Welcome to the diner.
Hey everyone, it's good to see you all again. I think this has been a much awaited topic for us to discuss. So today we're going to be really focusing in on the relationship between the gut and the brain and how this all fits together with Parkinson's disease. This is a big topic. And so right now we have this split at least into two visits. It may actually spread this out even more if the desire is there by all of you. The other thing that I'd like to do a little bit differently is, I want to try and go through this slowly. And so if we don't get to everything, no big deal. I'd rather you guys ask questions as we're going along, rather than kind of getting lost before we, move on to the next topic. So, unmute yourself, please feel free to ask questions as we move through this. I think this is a pretty important topic for us to move through. So let's get started. There. So I think for some people, it may be a surprise that is the gut actually involved in Parkinson's disease? And, for a lot of reasons, we know this to be true. In fact, just simply existing with some of these symptoms. So we know that constipation is probably the most common GI symptom that patients with Parkinson's experience. And again, this can be one of the very first symptoms that pops up, even decades before you actually get diagnosed by a clinician. But there are a lot of other GI symptoms that can come up with this as well. So nausea can be a big problem for some people, or something called early satiety. So people get full really fast. And this is just a quick look at some symptoms people experience with PD that are GI symptoms. On top of that, we know there are a lot of changes that can affect how your Parkinson's meds are working because of the gut. And we'll talk about that in a little bit more detail. And again, like these are things that can show up a decade or more before things like tremor and stiffness and slowness show up as more traditional Parkinson's type of symptoms. This can be the first thing that people retrospectively looking back, they're like, oh yeah, I did have a problem with that. Just again, show the strong connection between the gut and Parkinson's disease. And then top of that, and really why we're talking today is that there's been a lot more interest in this idea that maybe Parkinson's starts in the gut and not really necessarily the brain. And so we want to try and start unraveling that a little bit for everybody today, because this just a couple of years ago was a big topic in the news. I mean, it was a big splash. And I try to cut away some of the fat from understanding what this is going on about. So I think the key thing that we have to have some understanding of here is that there is a direct access between the brainstem and all of our internal organs. And this is primarily done through the vagus nerve. On top of that, our guts actually have nerves connected to them. This is how they know when to move, and if we need different nutrients, or if they need to put more water into your colon. And this is how this happens. And so many people call this nervous system, and so it's called formally the enteric nervous system, but some people will call it the second brain, because there's so many nerves that go into all of our internal organs. And again, the main highway of doing this is called the vagus nerve. So not Las Vegas, but the Vegas. So think of this as the internal highway that really connects information from your brainstem and back down to your gut. But this goes both directions. So this is the way that the insides talk to your brain. Now, this is really, really critical, because this is how things like digestion are managed and controlled by your brain. But this includes things like heart rate and how you're breathing, how you swallow, but other things like coughing and sneezing, and even hiccups. Hiccups are caused by irritation of that nerve. And so this is how we know that there is this direct connection between the brain and the gut. But we all know this personally. This is how things like stressful experiences and really urgent things that really worry us can cause bathroom emergencies or make you have to run to the bathroom with GI distress. This is how this is happening. But this also can explain why gut pain and other internal organ problems can cause people to have issues with like depression and anxiety. So again, this is a two-way street with the vagus nerve and the gut brain, the way it's put together. Any questions before I move on? Okay. So when I think it's really important to remember that there's a lot of nerves that go into the gut and all of our internal organs. And again, think of this as like a second brain. What's really important to know as well is that half of your dopamine is in your gut. And again, just really speaking to this idea of this strong connection, especially for Parkinson's disease. On top of that, and we've actually known this for quite a while, that the neurons that are in like the brain, like the nerve cells that are in your gut that have dopamine, they're just as susceptible to some of the same damage that we see in Parkinson's brain neurons as they are in the gut. So things like synuclein and other just damage to those cells, it happens the same in the gut as it does in the brain. There's actually a really old study, I think it was from the 80s or early 90s, that looked at patients who had been diagnosed with Parkinson's, but had like colonoscopy biopsies done, way back in the day. And when they actually looked at those biopsy samples, you could see synuclein up to a decade before they were actually diagnosed with Parkinson's disease. So again, just really giving us this clear idea that there is this really clear link between Parkinson's and the gut and synuclein. And this picture, what you're seeing here is actually synuclein inside neurons within our GI tract. Now, there's a lot of other links with the gut and Parkinson's disease, and specifically Parkinson's medications. So we know part of the reason, so Sinemet itself is actually a combination of two medications. So carbidopa and levodopa. That carbidopa is really there to make sure that enough of the levodopa, the active part, gets up to the brain. So by having the carbidopa there, it's making sure that it's not being broken down in your bloodstream and it's actually making it upstairs to the brain. Now, there are actually bacteria in our guts. We all have a ton of bacteria in our guts, and we'll talk about this a bit more in a minute. But some of those bacteria actually can use up levodopa as well, and that also then leads to less dopamine getting up into the brain, and so it makes the medicines a little bit less effective. And there's some studies that we're looking at actually kind of blocking the mechanism that some of these bacteria use to actually chew up the levodopa. And if you do that, you actually get boosts in levodopa levels and dopamine up in the brain. So this is actually one potential way of helping to manage Parkinson's symptoms, is by affecting the gut and I'll call it the microbiome. So this is all the bacteria together is called your biome, or your gut biome. And I think many of you may have heard that that's one possibility for treating Parkinson's symptoms. Now, one thing to keep in mind, and this is a day-to-day way in which the gut is impacting day-to-day symptoms of Parkinson's, is just simply how things get digested. So take a pill by mouth and it goes down into your stomach. It turns out that it's actually really a lot of protein that actually blocks the ability to absorb or soak up the levodopa from your stomach. So things like eggs, milk, protein, cheese, all these things that have high amounts of protein will prevent or block that levodopa from getting in. It turns out that the same channels, when you eat protein, the same way you absorb that protein from your meal is the same channel that's being used to take up levodopa from the stomach. And so the more protein that you eat, the more is blocking the ability for levodopa to get in. So this is not a big problem for everybody. There are some people for whom this makes no difference. And in fact, when they've done these studies, it's like 10, 30% of the Cinemet would be blocked this way. So it's not everything, but it does have an impact. And for some people, that 10 to 30% can be, make or break a good day from a bad day. And so to keep all of this in mind.
So doctor, if we eat a lot of eggs and protein and cheese, does that mean our levodopa isn't working as efficient as it could? Is that right?
So it will block some of it. And so there are potential ways of working around this. So you can see here, imagine all these little circles here, that's protein. And it goes into the cells of your intestine, and then it's absorbed this way into the bloodstream.
So if I cut down my cheese and eggs a little bit, it would work better for me.
It might. So here's what I would tell my patients. So sometimes what you do is you just change the timing of what you do. So for a lot of patients, not everybody, for a lot of patients, you wake up in the morning, you've had nothing overnight. That's a real rough time. And so the solution for that first dose in the morning is don't take it with food. Let it kick in so you get that first dose that really has a bigger impact. Or take it with like crackers or cereal, something that's carbohydrates that's easier to break down and doesn't interfere with this process. So that's one solution. Mm-hmm. One other way is if you're like high protein diets it's a big thing right now. So if that's the case, maybe you just need to up the amount of levodopa that you're taking. So this is the other way around this if it's not otherwise bothering you. Now, the other solution, because the timing again is really critical here. So we usually say 30 minutes before you eat. That gives the stomach enough time to digest it and help get it into the bloodstream. Or you wait an hour after you eat. The idea is that this allows the food to get through the majority of the stomach. And so it's now emptier and more available to take up more of the levodopa or Cinemet. And I get really worried about my patients that are altering their lives around their medications. I really think it needs to be the other way around. And you need to work with your doctors to really come up with a better way of making that happen. It's hard when you're on 8 doses a day. That makes this very difficult. And sometimes you do need to just sit down and plan it out, just so that you can have a life. I hate it when patients are struggling every few hours trying to figure out, do I eat? Do I not eat? What do I eat? It really need to work hard to see if we can come up with solutions that are livable, doable. Does that make sense?
Yes.
Dr. Amber.
5 times and I space it. But I can tell you
that's
wearing off.
Yeah. And that's hugely helpful in knowing how to time these things a little bit. And again, if the mornings are worse, then maybe you watch the protein more in the morning, or if the evenings are worse, watch more of the protein in the evenings. So but knowing this can help you make those sorts of plans.
Thank you. Yes.
Mm-hmm. And there's another question.
Yeah, I was trying to get a question in. I don't know if you can see me.
I'm only getting a few pictures in the snapshot, but go ahead. I can hear you just fine.
Oh, okay. Dr. Amber, my PCP prescribed sulfameth, trimethoprim. He prescribed an antibiotic for a possible UTI.
Yeah.
It's making me very anxious. And I'm totally not myself.
That's probably the UTI, to be perfectly honest. Sometimes the bacteria that cause someone to have a bladder infection, they actually secrete stuff that makes you anxious, and actually really, the in of itself makes people anxious or even confused or not feeling well. It's actually probably the bacteria itself, and maybe not just the antibiotic.
Okay. The reason we went this way is that I basically haven't been feeling good all of July and August.
I was going to say, actually, it's kind of linked with this whole story. You know, the weather can have a big impact on Parkinson's symptoms. And it's either too hot or too cold, so that the both extremes can really have an impact. And it's partly because of the autonomic nervous system. So the ability to adjust, you know, to these different temperatures can be affected in Parkinson's, and it can make it really hard.
Okay.
Were there other questions?
There was a question in the chat room. I find that not all proteins are equal. Are eggs considered dairy? And also, since eggs contain choline, isn't that good for brain health?
So this is the very first thing is, please eat protein. You have to eat protein to survive. And that's again why I'm saying, you know, make your meds work around you. And so yes, no, there are huge benefits to eating eggs. I'm just saying, try to adjust the timing and when you eat these things. I've heard that patients with like plant-based protein don't feel this as much. You know, so and some of this is just an individual thing, like you just have to figure it out for yourself. But again, it's, you know, try to make it work for you, not the other way around is my big takeaway.
Dr. Amber, I forgot to tell you, I have a deep brain stimulator.
Oh, okay. Okay. I still bet that the UTI is causing more of the problems. But if you were recently adjusted, you might want to talk to your doctor and see if you can get in to get seen. Maybe there's an adjustment that's needed.
Thank you.
Yep. Let me move on. Okay, so I'm going to hand this over to Dr. Victor Van Laar.
Okay, so well, you've heard us say that there are signs these things are showing up in the gut early, that we can see these things in PD. So what are we talking about, what we're seeing? And what is being seen in the gut, or some of those pictures that Dr. Amber showed earlier, was misfolded alpha-synuclein accumulations. So many of you are probably aware that alpha-synuclein protein accumulations are very much linked to Parkinson's disease, and where you find them in the neurons of Parkinson's disease brain. What's very interesting about alpha-synuclein, and has come out recently, is as alpha-synuclein gets expressed, if you overexpress it, like if the neurons start expressing too much of it, or they have a hard time getting rid of it, it'll start folding up in ways that aren't useful. And this will lead it to starting to glue itself together from these large aggregations. So alpha-synuclein ends up becoming the major component of what we call Lewy bodies, the protein bodies that we find in PD and Lewy body disease. But the really interesting research that has been coming out over the last 10 years or so is suggesting that alpha-synuclein doesn't just make Lewy bodies in the cell and sit there. It can actually cause other alpha-synuclein around it, alpha-synuclein that's perfectly normal, to form this more toxic form and start gluing together. And it could even move to neighboring neurons and cause the alpha-synuclein in those neurons to start gluing together. And in this way, alpha-synuclein has been compared to something that we call a prion. So a prion is a protein that can trigger other proteins to start acting bad. And an example of a prion disease that you've probably heard of is mad cow disease, which is where a specific protein in the brain of affected sheep and cattle can actually spread throughout the brain and cause other proteins to clump up and lead to neurodegeneration. So we're starting to think that alpha-synuclein, when it's in certain forms, can do this. Now, alpha-synuclein, it's a protein that your brain expresses. It's a protein that your brain needs to function normally. So alpha-synuclein isn't the problem. It's when alpha-synuclein, it's when there's too much of it and it's in the wrong shape. So, don't be scared of alpha-synuclein, be scared of alpha-synuclein that's not doing what it's supposed to do. It's like anything, when it's not doing what it's supposed to do, it tends to do bad things. But in this way, it could actually be spreading the disease through the body. And this is what gets us to the, can you go to the next slide?
I wanted to talk about the figure real quick.
Okay, go ahead.
So the little star-shaped brown things, that's basically the bad synuclein. So this is synuclein that's been folded in a way that it wants to clump together, so it makes it sticky. And so you can actually, the cells can actually spit out this bad synuclein, and then it can be taken up by another cell, taken inside. And then when good synuclein, like just regular normal synuclein, sees the bad sticky synuclein, it makes it sticky too. And so now it's all kind of clumping together, and then little bits get out, and so on and so on and so on and so on. And when you have, the whole nervous system connected, you've got highways going back and forth. And this is how it's this spreading is how we think there's this gut-brain connection as well with PD and synuclein moving around. I just want to make sure we understand. Does anyone have any questions about this? Because this is a really key topic to understand the rest of this. Just let me know. Okay, we'll move on.
Or me, you can let me know too.
Let us know.
I have a question.
Yes.
Yeah.
When you said they did colonoscopies and they found alpha-synuclein, when we have our colonoscopies done, could we have them look for that?
Oh. Actually, I didn't hear you say colonoscopy, so I was surprised because that's not what those images were from. Those images.
I know, but Amber mentioned earlier on that they found.
Yeah, so it depends. So not all like hospitals hold on to all of the biopsies for very long. So it just depends on whether or not they have them. And then is it possible to get that sample if it's still there and have it analyzed? Probably. Now, the other problem is that, especially depending on when it was taken and when things were started, there may not be very many cells that have synuclein in them. And then was that biopsy done in a way that you actually caught one of these cells that had synuclein? So just because it's not there doesn't prove anything. And so just going back and looking at just your one biopsy, probably not going to be very helpful.
Right.
It is helpful when you're looking at lots of people and lots of biopsies. This is how we're able to figure this out, kind of retrospectively.
But just like in the brain, not every cell is affected. So just certain cells and the chances of getting those cells. And even then, if you're a person that already has Parkinson's disease, what is finding alpha-synuclein going to really tell us? It's just going to kind of confirm this hypothesis. So where this is headed is like trying to understand where the disease comes from so we can predict it in people. So, one of the future things might be able to, like, doing some sort of biopsy or synuclein scan in younger patients that are prone to PD, so we can actually predict to see if they have PD and stop it at the gut level.
There's skin biopsies are doing the same thing, like the nerves, like in your skin that you can feel with. They also can collect synuclein. So it's actually easier to get a skin biopsy than a colon biopsy. But this is all being looked at. And so this could be, you turn 50 and it's time to do your Parkinson's screen. And so we do like a skin biopsy or some other easy way of, getting a body sample and doing it that way. So, I mean, you're hitting the nail on the head. This is where these things are headed. And again, hopefully soon we'll have some type of therapy that will allow us to stop it very early before there's any brain type of symptoms. This is the goal. And this is probably one way for us to be able to get there.
So one of the biggest questions many people have, I'm sure some of you have, is, well, what triggers the alpha-synuclein to start getting sticky and acting bad? And the long answer is we don't. The short answer is we don't know. That's why most cases of PD are still idiopathic. We don't know. But in
We're
the
idiots
lab,
and we don't know.
Yes. in the lab, we have tested things that can trigger alpha-synuclein to start becoming sticky and misfolded in models. So one example is environmental triggers. So certain pesticides like rotenone and paraquat, when it's fed to animals rather than injected like we normally would for a model, we actually see some of the gut alpha-synuclein starting to make these misfolds very early on in the animal models. And we actually do see it in the injected animals too, but it's the timing isn't quite the same. So it does seem that there are environmental triggers, certain things that can be eaten that can trigger this. Another thing that seems to trigger this is inflammation in the gut. So certain things like that may be related to IBS or just gut inflammation in general, seems to increase the expression of synuclein in gut neurons, and the increased expression leads to increased misfolding, leads to increased accumulation. There is also some evidence that an imbalance of the gut flora, the type of bacteria that you have in your gut, can trigger this inflammation or can trigger this synuclein overexpression and lead to the same thing as the environmental triggers or the inflammation. And there is evidence, there have been very large studies done that have found that PD patients have a different and very distinct balance of bacteria in their gut as compared to healthy counterparts. So what this really means, we don't know yet. We still have a lot of study to figure out what the right balance is. Because just because somebody doesn't have PD doesn't mean that their balance is right either. Could be the balance that causes Alzheimer's, or could be the balance that causes IBS. We don't know. So we need to do a lot of work to see can changing the gut biome fix things. Yes.
I had a question. Besides bacteria, when we talk about gut biome, are we talking about pH level?
pH levels, not so much. The pH level of the body is pretty well fixed. Life exists.
Even inside the bacteria of the gut, though, it can be a little bit different, but it's influenced by the bacteria.
So, I mean, when we're talking about the pH of the gut, it has to do with what the things in the gut are doing, not so much with what you're eating. So, and when we're talking about gut flora, we're not just talking about bacteria. You are right. We are talking about gut bacteria, gut viruses, gut funguses, like all these things are living inside of us in a symbiotic relationship. And, 99.9% of the time, they're doing us no harm. It's when things get out of whack. And it's much more likely that they're causing the influence on things like localized pH or localized inflammation than something that you're taking in. So, be careful with those things that say, oh, you've got to alkaline or acidify your body, because there's very little that you can do that actually influences your pH. Your body works really, really, really hard to keep the pH really close to physiological pH, which is around 7.4.
Because I was asking this because they always say with toenail fungus that your pH is out of balance. And then
same with Canada.
Yeah.
They say that, and I don't know if there's a lot of evidence to back that up.
There really isn't. It's unfortunately, it's somebody trying to sell you something most times.
And they're selling in probiotics or different kinds of bacteria to put in there.
Correct.
Well, that's absolutely correct, but that's kind of a catchphrase. It's kind of a to attract you. But that doesn't mean that the probiotics are bad. It just means that they're not using the right information to sell it to you.
Right. Okay. And that information, like not all Parkinson's patients have toenail fungus, and that's because there's a buildup of fungus in their gut. I don't know, but
doesn't always mean that. Okay. And I did want, because of what we were kind of talking about before, I mean, they are looking to see, if we look at the microbiome of your gut, are we able to diagnose or predict that you're going to get Parkinson's? I mean, this is another way of biomarker for us to identify this sooner. So, I mean, we'll see. Yeah.
Because it's just like almost everything when we're studying Parkinson's disease, we still have this huge chicken and egg question. Like, yes, we see the different gut biome in patients, but is that caused by having PD, or did it cause the PD? You know, so I say here that there are some studies where they've changed the gut biomes of lab animals, like they've given them the same biome that we see in PD patients, and it seems to cause development of PD-like pathology, but we're also talking about mice and rats. Those aren't humans. So, I
mean, at the very least, it's clear that there's something going on. There's enough smoke, right? There's got to be fire there. Yeah.
And that's where we're at with this. Is that you're going to hear the word hypothesis a lot in the rest of this talk, because these are things we think are related to PD, but there's still a lot of study to happen. So we can go to the next slide.
I just did this. Yeah,
you can talk
there's just so much. And, you know, this I guess also suggests why we need to be thoughtful about this. And this is also one way to really support this idea that most of your immune system's in your gut. And this is why taking good care of your colon and the rest of your intestines is so important for everybody's health, no matter if you've got Parkinson's or not. And you know, to keep this in mind as another, you know, it's the inflammation that can happen here. So you get, you know, inflamed cells, and maybe that's part of the cause. So this is also meaning, you know, good, healthy diet. That's the best way to support a healthy gut, is just good, healthy diet. And that means eat fruits and vegetables and exercise and water. All of that contributes to a healthy gut. And this is why we need to really pay attention to that, because most of your immune system is there. But I was just impressed at, I mean, how much bacteria is there. It's a lot.
So this is where we get into where all this science information has been leading, is, you know, where does PD start? So here about 10, 12 years ago, a scientist named Heiko Broc did a huge pathology study where he was trying to stage PD in the brain based on where there were alpha-synuclein accumulations. And then he did the study again, but he looked at more sections than outside the nervous system. And what he found was that the earliest signs of PD were starting to show up in the neurons of the gastrointestinal tract in the neuron. And that's where we first got this proposal that, you know, alpha-synuclein was acting like a prion, moving from the gut up the vagus nerve to the brainstem and olfactory system, and then up to the cortex. And the way he figured this out was that he was saying that it was corresponding with the patients reporting what we call the prodromal signs of PD, the things that happen 4 to 8 years before being diagnosed with Parkinson's. Things like constipation, unexplained anxiety or depression, losses of smell, sleep disturbances. These things were showing up in patients that hadn't been diagnosed with PD, but then when he was looking at the pathology, these patients had, you know, the ones who were reporting constipation had gut alpha-synuclein. The ones reporting anxiety, depression had gut and vagal nerve alpha-synuclein. The ones with loss of smell had olfactory bulb synuclein. And then he'd look at the patients that had just been diagnosed with PD. They had all of that plus now alpha-synuclein in the brain. So this led to this hypothesis, the gut first hypothesis, or also called the broc hypothesis, after the scientist Heiko Broc, that the dopamine, that the pathology starts in the gut, starts in the body, moves up through the organs by the vagal nerve, into the brainstem, into the brain, and then eventually, because it actually takes quite a few of the nigral neurons to die before you get the motor symptoms, and once the dopamine system in the brain becomes overwhelmed, then you see the motor symptoms. Oh, that's right, I can't control the presentation. So
yeah.
So these are some more examples of what Dr. Broc was seeing in some of the samples he was looking at. So from people that had passed away when they were reporting symptoms like constipation or anxiety, depression with constipation, he was seeing, a lot of evidence of these synucleinopathies, was what we call them, the accumulations of synuclein in, like these are all intestinal neurons that you see here.
All the dark brown stuff, so like these really dark, almost black looking things, that's the synuclein, and it's just choking up these nerves as it's coming through. That's what you were seeing here.
Mm-hmm. And then he would go on, and then in patients that were reporting the movement disorders, he would see evidence of synucleinopathies now in pink rather than brown. In the motor nucleus of the vagus, that's what that dorsal 9-10M nucleus is. Then he would see it in the brainstem, the locus ceruleus and the substantia nigra. He would see it in the cortex, in basal ganglia, the amygdala, and in the olfactory bulb. So this is a person that had diagnosed PD, had movement disorder symptoms, constipation symptoms, issues with sense of smell, issues with sleep, and they have synuclein in all of the places that would affect that.
And just to kind of emphasize again, it's this like upward movement of synuclein. So it comes up to the brainstem and then sort of forward. And this is, it looks like there's this, upward progression of synuclein by looking at it this way. Any questions on that before we move on?
We know what kills this. Does it stop? Once it starts, can you stop it?
That's a really important question. And I actually noticed we don't actually have a slide on this, but it turns out that there are some people that have really bad reflux, and it's so bad that they sometimes go and have a vagotomy. So they actually cut that vagus nerve. And same kind of thing. When they cut the vagus nerve, they say, gee, these people don't get Parkinson's at a much lower rate. What's up with that? So this was the paper that actually sparked this whole big, it was what, 2013, somewhere around there, that all of a sudden this big splash of like the brain gut axis, and there's this prion spread of synuclein. That's the paper that really triggered this. So this was one of the theories, like, why don't we just give everyone a vagotomy, which also doesn't make a lot of sense. Right. But the idea is that could you stop the spread from coming up this way and stop everyone from having Parkinson's? And I do not think that is reality. I don't think that will work. And again, and I want to emphasize this point again and again today, it's a two-way street.
Right.
So just like things can start in the gut, they could potentially start actually in your nose, like in the olfactory nerves, the stuff that makes you, how you can smell. It's the same highway. There are a few nerves that come right out of a brainstem, and those are the nerves that actually help your face move and feel. Just like the gut, the vagus nerve runs up this way to help you know where make your guts move and feel. These are the one way that your nervous system is directly connected with your environment. So no, I don't think that we should be snipping everyone's vagus nerve. I don't think that will eradicate Parkinson's. But you hit the nail on the head again. This is one of the theories, is maybe we could stop it for people who have gut first type of Parkinson's. And we have a slide on that coming up in a second. So, but here's more of the broc staging.
Right. So that's the part I want to emphasize, is that we're talking about one hypothesis right now. In this case, the gut first hypothesis, or the broc hypothesis. So this is actually where it started, was he was just looking at brain tissue to start with. And he noticed that the people that had premotor symptoms only had synucleinopathies. So like these broc stages, what we now call broc stages 1 and 2. So people that only had premotor symptoms, but clearly had evidence that they probably would have been diagnosed with Parkinson's or had very early Parkinson's. They only had synucleinopathies in their olfactory bulb and the brainstem. And then when you look at 3 and 4, people that have the sleep and motor disturbances, neither it's the entire brainstem, parts of the cortex, parts of the, like the amygdala, the thalamus. And then stages 5 and 6, very advanced Parkinson's that involves cognitive, function, emotional function. now the synucleinopathies are everywhere and the brainstem is completely overwhelmed with them, causing the severe motor symptoms. So this is where it started. And then when you look down, he said, hey, it's got to get to the brainstem from somewhere. Hey, it seems to be coming from the vagus nerve, which is coming from the gut. So, and then when you combine that with the epidemiological evidence that if you cut the vagus nerve, those people have a much lower rate of Parkinson's disease, that just turned into this huge, oh, everyone gets Parkinson's from the gut. The problem is scientists love to want to find the answer. And Parkinson's has shown scientists time and again, there isn't one answer. So in addition to the broc hypothesis, we also have others. So another example of this, that's actually an extension of the broc hypothesis, is combining, it'll be the next slide, unless you had something else to say.
I just again, just to show this, like it's like up and around type of distribution of synuclein. And the other thing I wanted to point out was, you know, this to me tells me there's multiple ways synuclein can cause problems. The Lewy bodies, or the big clumps of synuclein, they look different if they're coming from the olfactory bulb or like where your nose than they do in the brainstem coming from the gut. So it's just, there's not just one clean answer here.
So, you know, cutting the vagus nerve didn't mean that no one got Parkinson's disease. It was just a much lower rate. And also, synuclein isn't the only thing we see in Parkinson's disease brain. We also see evidence of inflammation. So then there's the broc hypothesis with inflammation. And the idea here is, yes, you get the synucleinopathies, they're spreading up the vagus nerve, they're causing synucleinopathies and Lewy bodies in the brain, but that's not the only problem, because we don't really have good evidence that synuclein alone causes neuron death.
Yeah.
And that's the most important takeaway here is we don't actually know that Lewy bodies are bad. We just know that they are in Parkinson's and body dementia brain. So what's killing the neurons? So the idea here is not only are you getting synucleinopathies in the gut, but they're also associated with inflammation in the gut. And that's triggering your, not your like antibody immune system, but what we call the innate immune system. So like killer T cells that come in and clean up inflammation. So inflammatory microglia and inflammatory T cells. And the problem is if they come in and they recognize synuclein as a bad thing and eat it, now they're going to start doing that all over the body. And that doesn't have to travel up the vagus nerve. So this is what we call, so the synuclein is traveling by the vagal nerve dependent pathway, but inflammation goes by a non-vagal nerve pathway, and that's all over the body. So in addition to the synucleinopathies, you're also getting inflammation happening all over the body that recognizes synuclein as a bad thing and starts trying to go clean up inflammation in other parts of the body. And it may be cleaning up the inflammation in the brain, and that may be causing those neurons to suffer. So
this goes back to this idea of maintaining good immune system in your gut to help keep that response down, because like these little microglia, they talk to each other, and that's how they say, hey, you know, I had this, you know, bite of this synuclein. It's bad news. Like, you need to know about this. And then it tells its neighbor and it tells its neighbor. And that's how that one spreads. And that's how it goes body wide to trigger the immune system that this is an issue. And there has been a lot of theories that maybe all neurodegeneration, so Parkinson's and Alzheimer's and ALS, all of them, maybe they all have an actual autoimmune component to them. And that's where this is coming from.
An autoimmune or an innate inflammatory immune response, you know, something that involves inflammation and the body's ability to clean up inflammation. So, and like I said, that travels by a non-vagal nerve pathway. So imagine if we just gave everyone, you know, vagotomies, but the problem is they've already got inflammation from gut alpha-synuclein. We may not stop anything if the synuclein is even started in the brain at all. So, and then that leads to the next hypothesis, is there are people that get Parkinson's that don't have any of those early symptoms of Parkinson's. There are people that get Parkinson's that don't have constipation, that don't have restless leg, that don't have sleep disturbances. So where that don't have anxiety and depression. So how did they get Parkinson's? And there's this huge study. I can't remember his last name. Borgman.
Borgman, yeah.
Borgman.
Borgman.
That looked at MRIs of tons and tons and tons of PD patients. And what they found was that they were looking at PD patients that had RBD, I cannot remember.
REM behavioral sleep disorder, yeah.
PD patients that first reported REM behavioral sleep disorder, or PD patients that did not report it as an early symptom. So these would be patients that, the thought here was, well, REM behavior sleep disorders are going to be controlled really deep in the brainstem before the substantia nigra. But if you're not getting that first, then maybe things skipped over that part of your brainstem. And instead, what they found was there may be two different types of PD. There may be the PD that starts in the gut. The synucleinopathies start in the gut, spread through the vagus nerve and lower brainstem. Those are the patients that report the RBD early, or in constipation, things like that. But then there's a whole nother set of patients that had the cognitive disorders, had sleep disturbances, had movement disorders, but not the sleep disturbances. And the thought is there that it's starting somewhere else, probably in the olfactory bulb, and then spreading backwards through the brain and brainstem and then down, and causing constipation and sleep disorders later in the disease. So this is another reason that just doing vagotomies may not work. There's going to be a whole subpopulation of PD patients that still exist because their PD started in the nose or in the cortex. You were going to say something?
Yeah, just the RBD, so the REM behavioral sleep disorder, just in case not everyone's aware. So this is like the moving, kicking, screaming, yelling out, really restless in bed, falling out of bed, hitting your spouse. That's RBD. Not everybody has RBD, but you're way more likely to have it if you have Parkinson's disease. This is another one of those symptoms that can show up way ahead of the motor symptoms of Parkinson's. And in fact, if you are someone who has RBD and no Parkinson's symptoms, the chances of you going on to get Parkinson's is like 90%. I mean, it's super high, or some other type of synucleinopathy is what we call it. So some other disorder where synuclein's doing the same kind of junking up and gluing together type of issue.
So the second I just want to finish this. Sorry.
Yep. Nope.
The other link they found was with PD associated with gene mutations. So gut first RBD positive PD was very often not involved with any kind of known gene mutation. However, brain first was very often associated with a gene mutation, suggesting that these neurons were just more susceptible to the gene mutation and that the PD started up there. So now I'm not saying that this is a division between environmental versus gene, because the words very carefully used here were more likely and less likely. But there's still evidence that the real takeaway here is that PD can start anywhere, but once it starts, it seems to want to spread. This becomes a whole body disease, and we have to look at it and treat it as a whole body disease.
I actually am expecting this theory to really take off. This is actually relatively new. I'm guessing this was just published in the past couple of years. And so I think this may help. Yeah, I think this may help us as Parkinson's doctors potentially to explain like what's coming next, or what's not going to happen, or maybe if it's gut first, you really focus on gut type of health rather than just more brain stuff. I mean, I think this will help us. And in truth, one of the biggest issues in trying to come up with new treatments for Parkinson's is because no two patients with Parkinson's look the same. And I think this is actually potentially one way for us to understand why there's just so much heterogeneity. So there's so much variability within patients that have Parkinson's. This is actually one way for us to help to start to understand that along with the genetics.
So before we move on to our wrap-up slides, because that was what we're going to talk about in this chapter. There was an excellent question. What exactly is inflammation?
Yeah, so that's a really important question. So I think inflammation is the best way to think of it is it's the immune system and how it's reacting and the stuff that it's making or producing to then communicate to other cells. So again, when you have like one cell, it takes a little bite of that synuclein, tries to get rid of the synuclein, and it realizes, ooh, this is bad stuff. And it's able to actually secrete signals to the next immune cell to say, this is a problem. And it's that secretion that it's doing that then spreads throughout the whole body, because now it's in the bloodstream. And so all the immune cells that in your bloodstream have the opportunity now to pick up that signal. And so it's this pro-inflammatory state, meaning that you've changed this immune system that's just kind of, bouncing along. And now you've triggered it, that there is a bigger problem. And now all the immune system is overreacting. This is where things like reactive oxygen species and oxidative stress and all of these type of terms start coming into play. So it's this overall state of the body that is primarily because of how the immune system's behaving at that moment.
So leading into the next question, which is, how does this relate to the Parkinson vaccine, which I did want to talk about. Because that's in clinical trials. So where we really have to delineate here is inflammation versus the immune system. Inflammation can lead to activation of the immune system. But when we're talking about inflammation, one really good way to think about it, and it would be a lot easier if I had a picture because you can tell really quickly, is when you have, let's say you have a cell that is kicking out something bad, like not one of the immune cells, but let's say one of your brain cells is sending out signals that says, hey, I'm dying. You can almost think of that as like a fish in the water that's struggling. And then all it takes is one shark, one microglia to come in and say, hey, this is bad. Suddenly it sends out signals, so all the sharks come in. And when you've got an inflammation at a spot, like the microglia just swarm like sharks in a feeding frenzy to that spot and can cause this massive inflammation. So like when your leg swells, when it gets hurt or infected, that's all your immune system coming, that's all your immune cells coming in and saying, hey, something's really bad here. Let's cause a fever to burn out the bad thing. Let's all swarm and eat the bad stuff. That eventually leads to your body producing new antibodies to eat up the bad stuff. Now, vaccines skip over that step. Vaccines take the bad stuff and put it right in your bloodstream, right where your antibody producing cells can see it. And they say, oh, here's this new thing. We'll start making antibodies to it. Now, rather than trigger this massive immune response that can kill off cells and cause a fever and everything, you've already got making the antibodies that target the bad thing, gets it eaten up, gets it out of the way. So the idea of the PD vaccine is we just skip over the step where your cells are already suffering from alpha-synuclein and triggering this shark feeding frenzy of microglia. You're just already making antibodies to alpha-synuclein. It goes in, it attaches, your immune cells go in and eat it out in a more precise manner. You don't get this massive inflammatory response. That's why vaccines work, is you don't have to get sick. You just get a little sick, and then your body is primed to make sure it doesn't happen again.
And I just want to kind of emphasize again, there is this very delicate balance between the immune system. You want it to respond. This is how it protects you from, getting sick from things in your environment. You just don't want it staying too active for too long. And that is part of what we think is happening in Parkinson's. It's hyperactive, and that's what's leading to it going overboard and now damaging the cells, as well as trying to attack whatever, like the synuclein, for example. And so it's trying to find that really fine line between the two states.
It's a balance. And that is one of the other problems with Parkinson's is alpha-synuclein is we're talking about a chronic disease. When you get sick from a virus or a bacteria, your body goes in, attacks it, makes antibodies, clears out, you're done. You get healthy. You start making an inflammatory response to alpha-synuclein. The problem is the alpha-synuclein is everywhere, and your body's still making it. It's still accumulating, and this just goes on, and you get this prolonged inflammatory response that doesn't go away, and then it just builds and builds and builds, and you start killing off neurons. And that's one of the hypotheses that we think can lead to neurodegenerative diseases.
It's almost like a feed forward. Like there's a certain threshold. Once you get past it, the body just starts overreacting, and then it's do that for long enough and do that chronically. And this is how Parkinson's just continues and continues and continues.
Now, can DBS stop the spread of alpha-synuclein? That's actually a really interesting question. There's actually some work trying to see if DBS can be a disease-altering therapy, not just
hard studies to do.
But those are really hard.
But this idea that Parkinson's, when we look at like the electrical signals in the brain in Parkinson's, they're different than someone who doesn't have Parkinson's. And in fact, sometimes I've seen this in the OR, like you can hear the cells that are behaving badly because they start firing at the same rate that people are having tremors. So they're just like there's the shaking, you can hear the cells firing the same way. So there's just a lot of ways of, can we change the electrical activity to slow that down to then affect the symptoms? And is that also going to maybe help then the inflammation and synuclein and all these things? So this has actually been of great interest for many, many, many years. It's a really, really hard question to answer well.
So that said, I want to put that on with the caveat that DBS is affecting a different population than what we are seeing die from the synucleinopathies. Because you're talking about the subthalamic nucleus as opposed to the dopamine neurons of the nigra, which are the ones that we are losing. So the synuclein's still there. It's already spread everywhere it's going to spread. It's just going to keep building up. So there's a lot of evidence. There's a lot of work to look at if this is happening, but like Amber said, these are tough studies to do, especially because DBS tends to be done in later stage PD patients. And some of what we've seen, honestly, hasn't really been very clear whether it's helping or not. We do know that DBS absolutely helps with the symptoms, and it's primarily seen as a symptomatic treatment. But there's a lot of work.
I think the question now is, could you change the way you're doing DBS or delivering the stimulation that can then lead to neuroprotection or disease stabilization? That's long been desired, but we don't know yet.
Yeah, long way to go. So we're right after 1 o'clock. So, and we just finished up what we wanted to talk about. So good timing. So for next week, so takeaway messages from this talk. Yes, there are clearly links between gut health and brain health. We know that. We've known that.
You can take nothing else away today.
Since the 70s, we've known this. But it's just been within the last decade or so that we thought, oh, maybe this has something to do with PD too. And there may be two ways PD starts. It may start in the gut first. It may start in the brain first. Now that may be genetically linked. That may be environmentally linked. That may be just
combo
happenstance. We don't know, but we have to take both into account. But the links do suggest that PD may begin, some cases may begin as synuclein buildup in the gut. This may be impacted by diet. It may be impacted by gut biome. And it may even be the key to treating PD or stopping people from getting PD in the future, if we can use this as biomarkers. So a lot of research being done on this right now, a ton and ton. And that's why you hear so much about it. So what are we going to talk about next time? We want to talk about what treatments are available, like what's being researched, what can you do, what are the scams? You know, just like the genetic scams, just like the stem cell scams, there are already places out there that say, oh, we'll analyze your gut biome and tell you what diet you should be doing. Well, we don't know. We don't really know. Is the short answer there. Is like, if we don't know, they probably don't know either. They're just basing it off of what they've heard. So, but we do know that there are certain gut flora that we find associated with PD, certain balances. So we'll talk a little bit about that. And can you modify your own? Yes, but, well, we'll just leave it at that. Yes, but, and we'll talk about the but next time.
I really, say it again, Donna.
Thank you very much.
Yeah. So food for thought, no pun intended, or totally intended. Think about other questions that may have been sparked by our conversation today and send them to Casey. And let's pick it up from there for the next one.
Yeah, I think we really want to make the next one a lot more question driven, because we want to make sure that you're all comfortable with the answers, that all your answers get addressed, or all questions get addressed, and we'll fill in the gaps. Best we can.
I'm an identical twin, so it'll be interesting next time.
Oh,
very interesting. Okay. I like
Wonderful. This was fantastic. Thank you so much. Wow. And we're excited for chapter 2. This is recorded, so I'll email the recording to everyone. And someone had asked, is your presentation going to be available, like, to view later?
I'm okay doing that, honey, if you are.
Yeah, we can send you a link, just like I did with David last time.
Wow, thank you. Well, everyone, thanks for joining. Thanks so much to the Van Laar doctors. And we'll see you on September 17th.
Thanks.
And based on early reviews, I think you ought to start preparing for part 3.
Okay, yes. I kind of had a feeling it was going that direction, so we're ready. Thank you, everyone. Have a great weekend. This was a pleasure, as always.
Thank you.
Okay, bye.
Thanks, everyone.